All right, let's talk about cancer. So cancer happens when a mutation occurs that controls cell division. And this mutation can happen at any of the checkpoints. It can happen in S phase when DNA is being replicated, right? So if a mutation happens, whether it's caused by the environment, like from UV rays from the sun, or it's caused by something that's genetic, this mutation that affects cell division is going to cause cells to not be able to stop dividing. So basically, they're going to be going through the cell cycle over and over and over and over and over again, even when they're not supposed to. And so they're going to keep going through mitosis and spend as little time as possible in interphase and then rush back into mitosis and mitosis and mitosis again and keep splitting. And that results in a cancerous tumor. So again, in a basic science definition, cancer is when cells cannot stop dividing. And it's a bad thing, and it can cause diseases, and it can spread across the body. And it's really, really lethal a lot of the times. So this is a picture of a cancerous tumor. As you can see over here, it is marked in a different color, and the cancer cells are dividing faster than they normally would in the regular cells and the regular tissue around it. So they start to cause this mass that is known as a tumor. And a cancer is different from a tumor just because tumors can happen, but they're usually benign and they usually don't cause problems, and they can be removed and then they don't spread. The problem with cancer is that cancer can spread, as you see over here, and they can get into your bloodstream and then spread to other body parts or other organs and then create tumors across the entire body. And then at that point, it's usually incurable. So simply, changes in chromosome number and structure are easy ways to recognize mutations and causes of cancer, or basically going through the cell cycle too many times. So there are types of mutations like deletions that affect chromosome structure by making a part of a chromosome deleted or disappeared and no longer there. So a gene is gone. There's duplication when almost entire chromosomes or chunks of chromosomes are doubled where they're not supposed to be. So having extra genes is also bad. And then there's also translocation, and then one more on the next slide. But translocation is when the section of a gene on a chromosome is accidentally transferred over to a different chromosome where it's not supposed to be. So a translocation is kind of weird because it's basically like taking somebody's gene from one chromosome, like say chromosome 1, and moving it to all the way to chromosome 21, where it's not supposed to be. And it doesn't belong there. It doesn't get read properly. And so when it's supposed to be read for the gene production, then the location is wrong and it's not going to get recognized when it needs to be recognized. The last one we're going to talk about is inversion. So a gene on a chromosome, instead of being this way, it's going to actually get flipped and be read this way. And then when it's read, it's either read the wrong way or it just cannot even be read anymore because the stop codon now is first, right? But it's actually backwards. So everything in the gene doesn't make any more sense when it's being read. So therefore, if it was a gene that was supposed to stop cells from dividing and duplicating, then it wouldn't be able to do that anymore. So all of these are super, super big chromosomal structure changes that can cause severe mutations that prevent cells from being able to go through the cell cycle normally, and that would cause cancer. So here's a picture that shows deletion, where you can see over here, chunk A on this chromosome is removed, and so therefore it's been deleted. You can see duplication. D and E originally were just once through, but if you duplicate it, then you have A, B, C, D, E, D, E, F, G, and it's duplicated, right? It's doubled. Over here, you have inversion, where it used to be A, B, C, D, E, F, G, but during the DNA replication, something went wrong, and on the chromosome now it reads A, B, D, C because they got flipped, E, F, G. And then translocation would be saying that chunks of genes on different chromosomes are being swapped to where they're not supposed to be. So originally, one of the chromosomes reads A, B, C, D, E, F, G, H, and the other one reads L, M, N, O, P, Q, R. Instead, the bottoms of them get swapped or translocated, and now it reads A, B, C, D, E, F, Q, R, and L, M, N, O, P, G, H. And for example, during deletions, you can have things like the Williams syndrome that comes out of it, where you get mental impairments, physical development issues. You can get reproductive issues that result when entire genes are lost, because sometimes they are really, really important. So that's our brief interlude about cancer, and you're going to go ahead and finish your video worksheet.